And now I'm going to, in a very summarized way, show you what we saw. You can see, read, and my PhD thesis if you want to get into a lot of detail. But here I'm just going to give you some of the key concepts. So we found that above the comb of the proximal region and underneath the comb, the distal region, there are different cellular processes taking place. So we found that the cells only divide above the comb, the cells mix above the comb, and above, underneath the comb, below the comb in the distal region, the cells also mix, and they mix in a random way. However, we never see a cell dying, extruded in the region. What it makes sense, because if you are pushing, if the cells were dying, you will not be able to push. And we studied that in wild type tissues. Yeah, here this is an schematic showing male wild type, and here is the comb, and here we have these two regions, and this is an schematic of the first dorsal segment. Here is an schematic of the females. The females, the flies don't have a sexcom, they have a very similar, if you see this top region, the bristles look fairly similar. Yeah, row of cells together and some single bristles that maybe later I will talk more about it. However, in the distal area, there is difference. So what we did is we label cells that are homologous. So and try to study what are the cellular processes taking place there to try to identify what are the unique process of the that belong to the cell sexcom rotation. Yeah, to know if the interchange of cell neighbors it happened only during the rotation or is a normal process taking place during the leg elongation. And also we try to see if the cell extrusion also only take place here in the sexcom rotation or can also take place here in females that they don't have sexcom. And this can give us an indication of what is the unique process that take place during the rotation. Also, we use as a control other region of the leg, this top region above the sexcom that have a low influence from the sexcom rotation and try to see what generally takes place during what are the cell dynamics taking place in that region. And interestingly, we found that the cells are interchanging cell neighbors and are extruding in both in females and also in the control region. And if you want, there is a paper published by one of our undergrad students that describing a lot of detail what happened in females, or you can also look in my thesis. However, what I wanted to tell you is that this process seems to be everywhere in the leg elongation, so seems not to be involved, seem not to be fundamental for the rotation. However, there are two processes that only happen during the rotation: the increase in apical cell area and the reduction in apical cell area. So these two processes are fundamental, are only found during the rotation. Again, as I said, so these two are the key processes: the increase in apical cell area and the reduction in apical cell area. And why we talk about apical cell area? As I said before, we are using as a marker the UV decoherence that allow us to see this apical region. And now I'm just going to summarize. I'm going to show you all these different steps of the data: the mixing of the cells or interchange of cell neighbors, the cell extrusion. But now I want to stay mainly with these key components of information, that is the increase in and reduction in apical cell area. What we did here is called a heat map of the changes in apical cell area. Here you can see three specific regions of the leg: the male wild type that have display the comb rotation, the female wild type that they lack of sexcom, and this control leg region. And we try to see what happened during the rotation, what happened with the changes in apical cell area. And we have in red the cells that are 3 micrometers in area, and the highest number will be higher than 12 micrometers. And here you see this region, the distal region, and in the distal region, you see that the cells in the beginning, most of them look like red, so they are very tiny, and some oranges. And this shows that tissues start very with cells that are very tiny, and later, at 36 hours, almost at the end of their rotation, the cells have sizes that are blue, yellow, and green. Uh, most of the cells are much bigger, so there is a huge increase in apical cell area. On the other hand, here you have a lot of variation in the cells. You have cells that are blue, cells that are green, and some of them that are yellow and red. However, at the end, you see that the region above the cone, the cells that are there, so this region that we measure, all the cells, they tend to be much smaller. And we found that they're significantly bigger above the cone, and they're statistically significantly smaller underneath the cone after the rotation. However, when we went and studied the same process in females that don't have rotation, we didn't find any change underneath the cone. In the distal region, there is not increase in area. And when we went and studied the cells above the cone, the homologous region, each one of these is a cell. We didn't find any particular change. So it seems that this process only takes place during the cone rotation, and in the control-like region, we didn't find any significant difference, as I said before. So again, this process, this increase in apical cell area and reduction in apical cell area, seems to be unique of the sex cone rotation. Now I'm going to show you the other cellular process that we also study, and I'm not going to go a lot into detail. I'm just going to show you how we did it. In case that you want to test or you are curious about some of these questions, you can find all these ideas in more detail in my PhD thesis or in some of the papers. So to test for cell rearrangement, what we did is we made horizontal lines. So we colored the cell. For example, here if we make horizontal lines of different colors. So we made vertical lines. Here you find in the initial stage of the movies. So green, yellow, orange, and blue. And after, in the late stage, we test if the lines remain still together or the cells are mixing or not. So this will be the initial stage. And these ones, we see that this represent the final stage. And this is a very easy way to test if the cells are mixing or not, and if they display a clear pattern in other tissues. And we were studying that in other videos. We found that the way that the cells interchange neighbors seems to be very organized. So when we test this idea, I'm going to show you a movie, but first I'm going to show you more or less what results found, so it's much clearer for you. We found that when we made these lines in the initial stage and later we follow the cells through time, we found that some of the cells actually rotate. Similar as the sexome rotates, those cells also rotate. We found that, so this is the example. We found that other cells, they go from one line into two lines. So instead of finding one green line, like here, we start finding two green lines. So the tissue narrows and but at the same time, the tissue elongates. And this is a process that we will talk later that is called convergent extension. And this seems to reproduce some of these convergent extension-like behaviors. And in our cases, we found that the cells are mixing because some of the cells are dying. Yeah, and so the line mix because of that. So instead of finding a single process, we find a combination of process. And let me show you that. So we made here, you see one line in purple, you see a second line. We try to make it as straight as possible. We make this third line and we make a fourth line here. And this is just an example. We made more lines here. For example, you can see a red one here. But just to make it clear, I just wanted to show you that. Now I'm going to play the movie times. So here again, now I'm going to play the movie, the different line. And if you see, I'm going to play it several times. So for example, this purple line, they go from one line into two. If you see this yellow one, some cell seems to rotate. This one form will rotate in both directions. There's a lot of things happening. So we saw some cells that rotate to the right. Some of them rotate in the opposite direction. Some of them actually went down. Another process that it was very interesting is how the cells died. Yeah, in this case, now we know that it's cell death, but at that time we just knew that they disappear from the epithelium. Yeah, and I'm going to show you an example. I'm going to play this movie, but first I'm going to show you explain this, and every time that you see in the movies that you're going to analyze these big squares, those are cells that we label and we were able to identify that they died. So here you will see all these cells that they will disappear during the rotation. And we encircle it in three groups just to make it easier to see. So as you can see, as the sexcom rotates, those different cells are dying, and you can see numbers because we try to count how many of those cells died and in which slide they die to know if we can find a pattern. Maybe you can think, what type of patterns do you think that they will describe? What will be the dynamics? Do you think that first the blue cells will die, and this will happen in this movie, or it will happen in all the movies? You can also think maybe the ones that are close to other bristles are more likely to die because there will be a pressure while the comb rotates, and as there is a bristle, maybe there is more pressure on those cells. Or maybe you can propose different another hypothesis. And now I'm going to show you in the next slide what we found. We analyzed three movies, and I'm going to try to simplify the patterns that we found. Now I'm going to show you the results of the spatial and temporal patterns or temporal patterns of cell extrusion. So the sequence of events of cell death. So for example, we hypothesize that one potential option is that maybe these cells died first because it's close to the comb. Then another cell will die second, and then the third cell died here. Yeah. So maybe there is a sequence of events. As the sexcom rotates, the cells start killing one, then the other cell, and then the other cell as the rotation proceeds. Another option, it could be the opposite. As I said before, maybe this cell died first, then maybe this one dies second, and this could be the third one. So instead of going in this direction, it could go in the opposite direction. And we were trying to see if there was any specific pattern of the sequence of event of extrusion. And you can imagine multiple patterns, and there could be many options. And we were trying to see if there was any specific pattern of cell extrusion. And here I show you what we found. As I said before, those processes are very heterogeneous. So here we compare the movie one a movie number two, and a movie number three. And in these three cases, they are wall type. Here, you can see the sex come here, the sex come here, and here you can see the sex come here. And in the numbers represent the sequence of event of extrusion. And what you see here is that, for example, this is the first one, the first cell that died. Then this is the second one, third one. And if you follow the pattern, you see that there is not a very clear pattern. In the second movie, the pattern is even weirder, very weird. And in the third movie, finally, it shows again a similar, not very organized sequence of, or at least not a clear pattern of sequence of extrusion events. So, as I said before, this is something that we found that it was pretty common, that there was these processes are very heterogeneous, and I can show you with the interchange of cell neighbors and with other processes that something similar is happening. But I don't want to make this presentation this longer. You can, if you're interested in these questions, I can show you later more movies, or you can look into that in my in my in the PhD in my PhD thesis.